It's my great pleasure to chair this very last session. And I'm sure we will have a fantastic start with Stacey Harmer, who will tell us all about the complicated interactions between light responses and clock responses, looking at sunflowers. Stacey, the floor is yours.
Okay, so thank you so much for the introduction. And thanks to Klaus, Christian, Mio, and Kiyoshi for organizing the fantastic meeting. I've really enjoyed myself. And in advance, I'll thank all of you for coming, because I know it's hard to stick it out to the last session. So I'm excited to tell you about work that my lab's been doing studying heliotropism in sunflower. I'm actually a circadian biologist by training, and we haven't heard much about clocks at this meeting yet. So I'll just mention that circadian clocks regulate many aspects of plant growth and development. And particularly relevant, I think, to us here today is that the clock modulates how plants perceive light, hormones, and then how cells elongate. And in fact, it's been published that many tropic responses are gated by the clock. And what I mean by gating is that if you take identical samples, but give them the same stimulus at different times of the subjective day or night, you can find different degrees of response. So this paper from Colleen Doherty's lab, they showed that root gravitropism, the plants showed a maximal tropic response when the stimulus came at the end of the subjective day, and a smaller response when the stimulus was given at subjective dawn. And my lab, looking at shoot phototropism in sunflower, we found that the plants were most responsive to a blue light stimulus in the middle of the morning relative to other times of the day. So time of day really matters in terms of how plants sense and respond to their environment. And with that in mind, I was really fascinated by this movie from Roger Hangartner's lab at this fabulous Plants in Motion website, showing a sunflower plant, tracking the apparent movement of the sun. So during the day, you'll see that the stem, apex, the stem, the petioles, the leaves, they all move to follow the sun, to incline the leaves to presumably get as many photons as possible. So that's well known. I'm sure many of you have seen this. But what's quite remarkable is that at night, the plant goes back to a neutral position. You can see here, and then it keeps on going. So you'll see that before the sun rises, the apex, the leaves, the stems, they're all bending to face east. It's as though this plant is able to anticipate both the timing and the direction of dawn. And so this, I think, is a pretty fascinating process. And we were really interested to study it. But first, we wanted to know, okay, it's cool, but is it important? What happens if you disrupt heliotropism in the plants? And unfortunately, sunflower is a fascinating system, but there's not molecular genetic tools the way you have in many other species. So we couldn't just make mutants. Instead, what we did is we disrupted heliotropism by doing an experiment where we manipulated the plants as follows. We grew plants in pots in the field, and you'll see these plants are facing west at the end of the day. If you leave them, if you take them up the pots and kind of shake them a little bit to give them some mechanical stimulation, you'll see they're still facing west. But the experimental samples, former postdoc Haga Batamian, rotated the pots 180 degrees such that the plants were facing east at sunset rather than west. Of course, the plants don't know that they were manipulated in that way. And so the next morning, these plants are now facing west facing west rather than east, like the controls. So Haggap went and did this every night throughout the growing season for these plants, just to see is growth affected by manipulating, perturbing this heliotropic movement. And he found, in fact, that there was a big effect on growth. So here we're looking at leaf area, and the rotated plants may have smaller leaves than the control plants. And we found the same thing for biomass, that there was about 10% reduction in biomass for the plants that were rotated. So this physical disruption of tracking had a negative consequence for growth. So we decided, all right, it's a cool process, but it's also important for optimal growth of the plants. So it's worth studying further. And we had a lot of questions. And our first questions were, how do they do this? How do they carry out this bending movement? And is the circadian clock involved? And so to answer these questions, we did some pretty simple experiments. First, we grew plants in the field, and we just put dots on the opposite sides of stems with a Sharpie, and then did time-lapse imaging and measured the growth of the opposite sides of the stem over time. And what we found is that the east sides of the stems were growing predominantly during the day and very little during the night. Whereas the west sides of the stems, we saw an opposite pattern. They grew some during the day, but they grew much more at night. And so it's this differential growth across this symmetrical organ that's responsible for the bending back and forth. And of course, you can think of a lot of reasons why you might have differential growth during the day. We've all heard a lot about phototropism, for example, but we were particularly intrigued by this differential growth at night when there's no obvious difference in the environment. So this is how movement is achieved, but how are these different growth patterns across this symmetrical organ generated? And is the clock involved? So to answer that question, we grew plants again in pots out in the field. Haggop measured the angle of the tip relative to the horizon, so the tip of the main stem. And you can see that at dawn, they're facing east. There's a gradual movement towards the west. And at night, it moves back again, such that it's got its maximal eastward inclination just at dawn. Very pretty. Haggop took these plants in pots and then moved them inside into a constant light and constant temperature. And he found that in constant light conditions, this back and forth movement would persist for several days. It eventually dampened out, but for the first few days, you can see that during that first day inside, the plant's moving from east to west, and then at sunset, even though it's inside, it changes the direction of movement and moves from west to east. The following dawn, it changes direction of movement and moves from east to west again. So somehow this plant seems to remember the timing and the direction of movement that it had in the field. So this and other kinds of experiments led us to conclude that indeed the circadian clock was involved in these solar tracking movements. So how does it work? Our first thought was that probably it's just daytime heliotropism, followed by some kind of a nighttime reaction reorientation, and maybe the clock is somehow gating one or both of these processes. So we wanted to test that. And again, we don't have mutants. We can't look at phototropin mutants. So instead, what we did is we took a transcriptomics approach. We first grew sunflower plants in growth chambers, exposed them to unilateral blue light. They bend quite nicely to the blue light, as you would predict. And we measured stem bending angles over time. And we did a relatively long time course, and you can see maximal bending of these stems is occurring about 8 or 9 hours after the light stimulus. So what Chris Brooks, a former student in the lab, did is he collected samples from the lit and the shaded sides of these stems. This is from the first true internode of these plants. This is not the hypocotyl. Extracted RNA from these strips, epidermis, cortex, and some vasculature. And then we did time course and did RNA-seq. Okay. So Chris found two main classes of genes were differently expressed in these plants. At first, we got the first class you'd totally expect. We found rapid upregulation on the shaded sides of stems without much change on the lit sides of stems. And when we looked at Go enrichment for the genes in those categories, we found all the things you would expect. Things that promote growth, auxin signaling, response to gibberellins, brassinosteroids, response to light. And these were, many of the genes were things that were previously found in similar experiments done in brassica, for example. So we felt comfortable calling these phototropic genes. He found a different interesting class of genes that were up late on the lit sides of stems. And the sharp-eyed among you, when we looked at Go enrichment, we found those were enriched for response to auxin and light. And you may have seen on this bending time course that there's a little bit of bending back of the tip away from the light source at a late time point. And that's occurring soon after we see upregulation of the genes on the lit side. We think this is a reflecting auto-straightening or proprioception of the stems. So we were calling these genes auto-straightening genes. Okay, so how are these two types of genes expressed in heliotropic plants grown in the field? So we did our favorite experiment again. We took plants growing in the field, collected the east and the west flanks of stems over a 48-hour time period, did RNA-seq, and looked at expression patterns in those samples. And first, we looked at the genes defined as phototropic in a growth chamber. We found in the field that they, on average, were somewhat differently expressed across the heliotropic stems, but it was nothing like the manyfold differences that we observed in a growth chamber in phototropism. Instead, we saw relatively modest differences in amplitude. So they were differently expressed, but not at all in the way that we had predicted they might be. And then we next looked at the genes we defined as autotropic or proprioceptive genes. And it was a similar story. We saw differences in waveform in the field-grown plants, but nothing like the manyfold differences in expression we found in a growth chamber. So a little bit of a surprise there. And why are these, if these patterns are different as they are, maybe we should be looking at different kinds of genes in the field, not the things defined in a growth chamber. So what kinds of genes are strongly differently expressed in heliotropic plants? We found that most genes, in fact, were very similarly expressed across the stems. For example, this is you can see they're superimposable, really. We see this is a clock gene, LHY. It has the same pattern on the east and the west sides of stems. And we see the same thing for this clock gene, L3. And so we know now from these kinds of data, we conclude that the stems, the east and the west sides of the stems are not exhibiting different circadian phases. You don't see different growth patterns because one side of the stem thinks it's on Hawaiian time and the other side thinks it's on California time. The whole plant knows it's in California. So there's got to be something else. And when we did differential, just compared gene expression at individual time points across the stem, we found relatively few genes differently expressed in the morning, a few hundred differently expressed towards the end of the day, but many thousands were differently expressed across the stem at night. So that was a surprise. There's more differential expression at a time when the light environment is not different across the plant than at times when the light environment is different. But recall that there's very different growth patterns at night. So we believe a lot of these differential expression genes are reflecting differences in growth. For example, we find enrichment for cell wall genes in those groups. But when the types of genes that are differently expressed across the stem, they tend to have two main kinds: large differences in amplitude, as shown for this transcript, or differences in daily phase, as shown for this PIN4 homolog. And so amplitude and phase are different for some kinds of genes. And we then wanted to look at different times of day, what kinds of pathways are enriched for differential expression. And we were kind of pleased to see that at night, we found an upregulation of auxin responsive genes on the west sides of stems. Recall, the west side grows faster than the east side at night. So that made some sense. But we didn't see that at all during the day. We did not see enrichment for auxin terms across the stem during the day, which was counterintuitive because we had expected to find phototropic type genes strongly differently expressed at that time, but we did not see that. All right, so this analysis was suggesting a role for nuclear auxin signaling perhaps during the night, but not during the day, kind of backwards. So, okay, we don't have mutants. We can apply exogenous auxin to the plants and see if that perturbs tracking. So we did that instead. And we did this by dusting off some classic kind of techniques, putting auxin in a lanolin paste and just applying the paste to the opposite sides of the stem. So first off in this, I'll show you what would happen when you apply auxin on the east sides of stems at dawn. And you'll recall that during the day, the east side grows faster during the west. You might predict that applying auxin at dawn would reinforce that normal movement. And that's indeed what we saw. And if we did the same thing, but applied auxin on the west side at dawn, you might predict that would perturb the movement. And that's exactly what we saw. We saw a rapid response. And we got this result whether we used 2,4-D or IAA. We saw very similar effects. Okay, so that makes some sense. Artificial auxin gradients do disrupt daytime tracking, even though we don't see a transcriptome signature for phototropism at that time. However, what if we apply an auxin gradient at dusk? All right, so start out by applying auxin on the east side at dusk. Remember, the west side normally grows faster than the east at that time. Does that mess up daily movements? Actually, it did not. It had no effect until the following morning. So that was strange. And we found the same thing when we applied auxin on the west sides of stems at dusk. We would expect that would promote nighttime movement. We didn't see that. We only saw an effect the following morning. It's as though the plants were blind to that auxin application when it was applied at dusk, but they were happy to perceive it in the morning. Okay, so how could we explain this? Is it simply that the clock or some other process is influencing the plant's ability to perceive auxin when it's applied at dusk? And so to test that, we took a transcriptomics approach. We sprayed auxin at dawn, waited an hour, collected samples from the two flanks of the plants, did RNA-seq, did the same thing at dusk. And again, we were completely astonished because we found that plants treated at dusk showed an appropriate, what we'd expected as an auxin response. Many hundreds of genes were rapidly, mostly induced, but some repressed. But the same treatment at dawn, we actually found really very few genes were differently expressed. And so this is actually exactly the opposite. I would have predicted that you would see a strong transcriptional response at dawn and a limited response at dusk, but that's not what we found. So this did not solve our problem. It didn't explain why we don't see a growth response when we apply auxin at dusk. Okay, we thought, all right, let's go downstream of the transcriptional pathway. And so I'm sure all familiar that the proton ATPases are responsible for acidification of apoplasts and causing plant growth, and that these are regulated by phosphorylation. And it's now work from many labs has shown that the transcriptional tier 1 AFB auxin receptors can induce expression of sour proteins, some of which inhibit activity of phosphatases that will remove a phosphate group from the penultimate threonine that promotes activation of the proton ATPase. You can bypass this system by treating plants with a fungal toxin called fusicosin, which promotes the binding of 14,3,3 proteins to the pump and promotes pump activity. So we decided to try that same application experiment, but this time to use fusicosin rather than auxin, so that we didn't have to worry about what was going on transcriptionally. So Chris Brooks did these experiments, and he applied auxin or FC to the to plants at dawn. He did this on the west sides of stems that we predicted should disrupt the daytime movements because it's would force more growth on the west sides. So that's exactly what we found. However, when he did the same thing at dusk, he got the same result with FC that he got with auxin. That in this case, he's applied the compounds on the east sides of stems at dusk. That should counteract the normal greater growth on the west than the east. It should disrupt tracking, but we don't see that. We don't see an effect until the following morning. So still, there's a very time of day dependent effect on the plant response to that chemical treatment. Okay, so how to explain this? Seems like that insensitivity to auxin at dusk, it's independent of nuclear auxin perception. Could it be due to changes in the post-translational modifications of the proton pump itself? We know, so we don't have genetics, but we can do omics. These stems are nice and big. And so what Chris did this time is he harvested samples on the east and the west sides of solar tracking stems and did quantitative phosphoproteomics in collaboration with Justin Wally's group at those 5 time points. And when we looked at the number of peptides that were significantly differently phosphorylated across stems, we found hundreds that showed differential phosphorylation during the day, but actually thousands differently phosphorylated at night. A little bit reminiscent of what you saw for gene expression, that the differences at night were more dramatic than the differences during the day. So lots going on for differential phosphorylation. What do we have when we look at Go enrichment? When we look at the peptide, the proteins that are significantly differently phosphorylated across the stems, when we looked at the things that were enriched for more phosphorylation on the east side during the day, the one term that came up was regulation of intracellular pH. And at night, for things that were more significantly, more abundantly phosphorylated on the west side at night, we found regulation of intracellular pH. So that was exciting. And we went on to then look at the patterns of peptide phosphorylation for the proton ATPase. And what we found was that for this one particular phosphopeptide that corresponds to threonine 881 of AHA2 and Arabidopsis, we found no differential phosphorylation 3 hours after dawn. That's the time actually when the stem is not growing, it's not bending either way. At 8 hours or 13 and a half hours after dawn, when the east side is growing faster than the west side, we find that that phosphopeptide is more abundant. And conversely, at night, at points 17 and a half and 22 hours after dawn, we found that the phosphorylated peptide is more abundant on the